Hello everyone and welcome to the movie on the heart. One of the most amazing organs that we have. We're going to look first at the layers of the heart wall. So this figure shows us the three layers. The innermost layer is called the endocardium, which we see right here. It provides a smooth surface for blood flow and it's continuous with the blood vessels. You can see it's a layer of simple squamous epithelium. Then the thickest layer is the myocardium. That's the muscle layer of the heart, and that's the part that contracts to pump blood. And then the outer layer is the epicardium, and it forms the pericardial sac, which allows the heart to move freely. This space here is filled with fluid, and that's what allows the heart to move freely. The muscles in the heart are arranged in bundles, so there's a bundle around each atria and there's a bundle around the ventricles. This allows the heart to work as a pump because they all contract at the same time and work together. So the heart is a good pump. Cardiac muscle cells look like this. They have a single nucleus. They branch, and we can see better in a drawing. They connect with each other at what are called intercalated discs. They contain sarcomeres, as you can see here, just like the skeletal muscles. They use aerobic metabolism, which means they have lots of myoglobin, which you'll remember is an oxygen storing molecule, and they have lots of capillaries bringing them oxygen. So they have lots of myoglobin and also lots of capillaries. And if we look closely at the junction, we see the cells are connected by desmosomes here, and they are electrically connected by gap junctions. So the gap junctions allow for the electrical connection, and this allows the heart to function as one unit, as we can see in this video. The atria contract together and the ventricles contract together. Pretty amazing that we can see a human heart beating inside the body. These are individual cardiac muscle cells. They have what is called autorhythmicity. So these are cardiac muscle cells that have been isolated from mouse hearts, and they're in a petri dish all by themselves, and they just know they're supposed to contract. So they generate their own action potentials, and we'll show how that happens in a bit here. The heart is located here in a cavity that's called the mediastinum. It's suspended within the pericardial cavity, so it's surrounded by fluid here that allows it to move freely. And if we have any damage to the pericardium or the chest wall, fluid will build up in there and stop the heart from working right, and that's called cardiac tamponade. The heart contains one-way valves that control the direction of blood flow. So you can see the arrows there showing blood flow through the heart. The atria are going to send blood into the ventricles, and the ventricles are going to pump blood out to the body through the aorta. If we look at a cross-section of the heart through the ventricles, we see the right side is much thinner than the left side. That's because the right side only has to send blood to the lungs, and the left side has to send blood out to the whole body. So that's why the left side is thicker. It has a lot more work to do. And this shows the relaxed heart, and then when the heart contracts, both ventricles contract at the same time. So the inside of the ventricles is actually the same size. It's the thickness of them that differs because they have the same amount of blood in each side. When the ventricles are relaxed like this, the AV valves open, and blood will flow into the ventricles. See here, blood's flowing into the ventricles. When the ventricles contract, now the ventricle's contracted, the AV valves close and the semilunar valves here open, and blood flows into the aorta and the pulmonary arteries. And we have these chordae tendineae and the papillary muscles here. They prevent the AV valves from opening back up into the atria, and they prevent blood from going backwards. Our heart valves are really amazing. They can open and close about 100,000 times a day. So, and think about how many days you've been alive. Your heart valves have opened and closed a lot, but they don't break or wear out. Well, sometimes they do, but not usually. No man-made materials can do that. We can make prosthetic heart valves. They use materials from pigs, but they generally only last about 10 years. Here's our pig valve. Next we want to talk about atherosclerosis. You've probably heard of this. Here's a blood vessel with atherosclerosis, and you can see that it probably used to have this much area for blood to flow through, but there's lipid deposits in the tunica media, and that's called an atherosclerotic plaque. And you can see now there's not very much room for blood to flow through. And here's an actual blood vessel, and you can see the atherosclerotic plaque forming there. High cholesterol, high blood pressure, and smoking all increase the risk of this. And these plaques will build up in the artery walls, and they block blood flow, and that can lead to heart attack or stroke. This can be corrected by coronary bypass surgery, where they take a blood vessel from somewhere else and graft it into your heart to go around the blockage. Or sometimes they can do balloon angioplasty, which is what we see here. They stick this catheter into your blood vessels, and they'll take it into the vessel that has a blockage, such as one like this, or like this, an even worse one. And then they blow up this balloon, and that presses against the plaques, and it opens up the artery so that blood can flow easier. Often when they do this, they'll add a stent too. That's like a wire mesh thing that keeps the artery open. This is angiography, which measures blood flow in the heart. On the left you can see normal, and the right you can see someone with coronary artery disease. That is partial or complete blockage of coronary circulation. And if you don't give your heart enough blood, it's not going to work. So you'll have a heart attack and die. And this shows a artery with a stent in it that's been repaired to keep that blood vessel open, so this person won't have any more heart attacks. Next thing we need to talk about is the cardiac cycle. The cardiac cycle is all the events that are associated with blood flow through the heart. So blood flows first into the atria, then into the ventricles, and then out these big arteries. Systole, if you don't know, systole means contraction, and diastole means relaxation. So there's an atrial systole at the start of the cardiac cycle, followed by atrial diastole, and then after about 100 milliseconds there's a ventricular systole or contraction, followed by ventricular diastole or relaxation. Now, blood flows in the heart due to pressure changes, and the pressure and volume are inversely related. So we can change the pressure by changing the volume. And since the heart is a muscle, we can change the volume of the different chambers, and that's how we move blood through the heart. And each heartbeat is followed by a resting phase where the heart relaxes and fills. Now the atria contract first. So here's the start of our cardiac cycle. The atria are going to contract first, and that pushes the blood into the ventricles. then the ventricles will contract, and that pushes blood into the arteries. So at the start, we see here all 4 chambers are relaxed, and the ventricles are partially full. Then the first step is atrial systole, so the atria contract, and that forces more blood into the ventricles. And then atrial diastole, we can see, starts right here, and that continues until the start of the next cardiac cycle. Then we have ventricular systole, so the ventricles begin to contract. That increase in pressure closes the AV valves, so we can see the AV valves are closed here. And at this point, all 4 heart valves are closed. This is known as isovolumetric contraction. And as soon as the pressure in the ventricle exceeds the pressure in the arteries, the semilunar valves, both the pulmonary and the aortic semilunar valves, are going to open, and blood is forced out of the ventricles. This is known as ventricular ejection. And then the ventricles relax. We start ventricular diastole. And as the ventricles relax, the pressure drops and blood flows back against the semilunar valves and closes them. And then we have a period known as isovolumetric relaxation, where all 4 valves are closed and the ventricles relax, and this decreases the pressure. Then in late ventricular diastole, all the chambers are relaxed and blood flows passively into the ventricles because the pressure in the ventricles is the lowest at this point. And this cycle just keeps on repeating over and over and over until you die. So we're going to look at it next. This next chart shows us the pressure changes that happen in the heart. So the red line is showing the pressure in the ventricle, and we're looking at just the left side of the heart for this. The blue line shows the pressure in the left atrium, and the black line here shows the pressure in the aorta. So we'll start here. At the start, the pressure in the ventricles is lower than the pressure in the atria or the aorta. So at this point, all 4 valves are open. Both the AV valves and the semilunar valves are open. Then at this point, the ventricles start to contract. So first, this little bump we see right here is due to atrial contraction. We can see that atrial contraction has little effect on the pressure in the ventricles because the atria aren't very strong. But when the ventricles contract, ventricular pressure increases above atrial pressure. So at this point right here, that's where the AV valves close. And then when the ventricular pressure gets above the aortic pressure, that's when the aortic valve opens. And so during this time, blood's flowing from the ventricle into the aorta. Then once the pressure in the ventricle drops below the aortic pressure, so as blood leaves the ventricle and the ventricle relaxes, the ventricular pressure drops. And when ventricular pressure falls below aortic pressure, the aortic valve is going to close at this point right here. And then ventricular pressure continues to fall, and when it drops below the atrial pressure, the AV valves are going to open and blood will flow into the ventricle, and then it's going to repeat again and again until you die. And down here this shows us when the heart sounds are made. The heart sounds are caused by valves closing. So here's the AV valve closing, and then here's the semilunar valve closing. That's why it makes those 2 sounds, the lub-dub. Alright, next we're going to talk about cardiac output. Cardiac output is the amount of blood the heart pumps in 1 minute. And it is determined by the heart rate, and the stroke volume. So to figure out cardiac output, just multiply heart rate times stroke volume, and hopefully you'll get an answer in milliliters per minute. And we can change the heart rate or the stroke volume to change the cardiac output depending on our body's needs. So for example, when we're exercising, we're going to increase both our heart rate and our stroke volume to increase our cardiac output. And when we're sleeping, those are both going to drop. Next, we're going to talk about the heart's electrical conduction system, which is very amazing. So we'll look at how the electrical signal travels through the heart. Here's the entire system. But the electrical signal starts at the SA node, which is known as the pacemaker. And if that doesn't work now, we have artificial pacemakers we can give you, implant in you. But most people use the one in their SA node. The impulse is going to travel throughout the atria, spreading from cell to cell. And then it's going to travel next to the AV node, stands for atrioventricular. And then at the AV node, there's going to be a little bit of a delay, and that ensures that the atria contract. They finish their contraction before the ventricles contract. That ensures that the ventricles will be full when they contract. And then these impulses are going to travel along the AV bundle and then to the right and left bundle branches and then to the Purkinje fibers, and the Purkinje fibers are going to spread the signal throughout the ventricles. And also we have this moderator band here, which kind of is a shortcut, and it goes directly to the papillary muscles so they can contract before the ventricles do, and that gets them ready for the contraction, and they keep the AV valves from opening up the wrong direction. So next we're going to review action potentials. You learned this last semester, how an action potential happens in a neuron or a muscle cell. It's a little bit different in a cardiac muscle cell, but it's mostly the same. So just review, we start out at a resting potential. Then we have depolarization, which is caused by the opening of sodium channels. And then at this plateau, the sodium channels are going to close, and the potassium channels, they actually open the same time as the sodium channels, but they take longer to open. So as the sodium channels close and the potassium channels open, the cell repolarizes, and since these potassium channels remain open longer, we have what's called a hyperpolarization, and then the sodium-potassium pump works to bring things back to the resting potential. So here it is in words if you prefer that. When the cell reaches threshold, voltage-gated sodium channels open, positive ions rush into the cell. This is the depolarization phase. Then those sodium channels close and voltage-gated potassium channels open. This is the repolarization phase, and then the cell returns to rest. And this shows the action potential and the contraction, and they're on the same time scale for a skeletal muscle. And the blue is the absolute refractory period, and the orange is the relative refractory period. So you can see it's very quick. This is 100 milliseconds right here. So the action potential takes maybe 5. And then you can see the contraction is finished within 60 or 70 milliseconds. Now if we compare that to cardiac muscle, it's still very quick. This time is still in milliseconds. But you can see the action potential lasts much longer. Here it's like 5 milliseconds. Here it's like 250 milliseconds. And you can see the tension is generated more slowly, but it also lasts longer. Instead of this contraction in skeletal muscle lasted maybe 20 milliseconds, this contraction is going to last almost 300 milliseconds. And the reason the heart has this long, and we can see in blue this is the absolute refractory period, and then orange is the relative refractory period. The cardiac muscle has a really long absolute refractory period, and that ensures that the heart will rest. If the heart muscles don't rest, then the ventricles don't have any time to fill up with blood, and then your heart is contracting, but there's nothing in it, so it's really not doing anything. So that's why we have this long absolute refractory period in cardiac muscle. Now this shows us the stages of action potential in a cardiac muscle. First, there's a rapid depolarization, and this is due to fast sodium channels. And then they have a plateau phase. We didn't see this in neurons or skeletal muscles, but in cardiac muscle we have this plateau phase. It's due to slow calcium channels, and this is what gives them a relatively long refractory period. It's still only 200 milliseconds, but that's much longer than it was in skeletal muscle. And as the slow calcium channels close, potassium channels open, and the heart repolarizes. And I showed a video of heart muscles beating by themselves, and we call that autorhythmicity. And they're automatic due to what are called funny channels. These are slow sodium channels that open when the cell is repolarized. So as the cell repolarizes, these funny sodium channels start opening. Normally the sodium channels open at threshold, but these are special sodium channels that we only find in the heart, and they open way down here at a lower voltage. And they generate what's called the pacemaker potential. So as soon as the heart relaxes, it starts to depolarize again. The autonomic nervous system can alter the heart rate. This shows what happens with parasympathetic stimulation. It's going to open potassium channels, which causes a slower depolarization. The open potassium channels is going to lower the resting membrane potential down, and it's going to take longer for the heart to generate a new action potential. So that slows the heart rate. Whereas sympathetic stimulation is going to open some sodium channels, and that's going to increase the resting potential. So the heart doesn't have as far to go to reach threshold, and so it's going to reach threshold faster, and that's going to increase heart rate. So that's how the autonomic nervous system changes our heart rate, and that's the main factor influencing our heart rate, is the autonomic nervous system. So normal heart rate is between 60 to 100 beats per minute. Slower than 60 is called bradycardia. Faster than 100 is called tachycardia. Now that's resting heart rate. Obviously when you're exercising it's going to go up, and that's fine, that's normal. This is all controlled automatically in the medulla. The cardiac centers in the medulla control our heart rate. We're going to talk next about stroke volume. Stroke volume is determined by the end stroke volume and the end diastolic volume. So remember, the cardiac output depends on the heart rate and the stroke volume. So this shows kind of an example of using this chamber as our heart. The end diastolic volume, that's as full as the heart gets, and the end systolic volume, that's as empty as the ventricles get. So the end diastolic volume is influenced by, we use EDV to abbreviate for end diastolic volume. It's determined by the venous return, which is basically how much blood is coming back to the heart, which is influenced by pretty much our exercise. The filling time, which is how long the heart waits in between contractions, and then the preload or the stretch of the heart. So the venous return is increased by things like skeletal muscle contraction. We learned in the last chapter how the veins have these one-way valves, and there's not a lot of pressure in the veins, so they rely on skeletal muscle contraction to squeeze them, and that sends the blood back towards the heart. So venous return is increased by skeletal muscle contraction, and it's decreased by things like blood loss. The filling time is determined by the heart rate. So if the heart rate slows down, we have more time to fill, and that will increase our EDV, our end diastolic volume. And then the preload, or the stretch of the heart can contract more forcefully if the sarcomeres are at an optimum length, just like skeletal muscle contracts best at an optimum length because you have the maximum overlap of myosin and actin. Then the end systolic volume is influenced by the contractility and the afterload. The contractility is just the amount of force generated when the heart contracts, and so more calcium is going to give us more contractility. Greater contractility is going to give us a smaller ESV and systolic volume, because if the heart is contracting more forcefully, it's going to pump out more blood. Afterload is the amount of force the heart needs to produce to open the semilunar valves. And anything that restricts blood flow is going to increase afterload, increasing the end systolic volume. So if there's more resistance in the blood vessels, that's going to increase the end systolic volume. And cardiac output changes on our bodies depending on our body's needs. So resting cardiac output is around 6 liters per minute. But when you get into cardiac exercise, it's going to increase to 20 or 30 liters per minute. For trained athletes, it can get up to 40 liters per minute if your heart and your blood vessels are all in great shape and you have lots of blood. So to change the cardiac output, we mentioned before, we can change either the heart rate or the stroke volume. So if you get that question wrong on the test, I don't feel sorry for you. The main things influencing the heart rate is autonomic nervous system stimulation, temperature, and hormones. And things that affect the stroke volume, one thing is the resistance in the cardiovascular system. So that resistance comes from the vessel diameter. Something with a smaller diameter is going to have much greater resistance. So you can see here a diameter of 2 centimeters has a resistance of 1, a diameter of 1 centimeter has a resistance to 16. So decreasing the diameter by half increases the resistance 16 times, and we don't need to worry about the math of that. Just know that a smaller diameter means greater resistance. Vessel length also increases the resistance. So a longer blood vessel is going to have more resistance than a shorter blood vessel, but it's not near as much effect as the vessel length. The vessel diameter. That's the main thing. And another thing that affects it is the viscosity of blood. Viscosity is like stickiness. So here's the viscosity scale. Water's down here, water's here. Blood is up here about 5 times thicker than water. Maple syrup's about 10 times thicker. Molasses is 300 times thicker. So blood is thicker than water, but not by much, and that leads to some of the resistance. Blood flow depends on arterial pressure and resistance from the blood vessels. And as blood flows into smaller arteries, arterioles, and capillaries, the resistance decreases. So this figure here shows us the area of the blood vessels here, the blood velocity here, and the blood pressure. And you can see as it gets into the smaller blood vessels like the capillaries, the resistance decreases and the velocity decreases. And blood velocity here is slowest in the capillaries, and that's a good thing. That's where nutrient exchange happens, so we need the blood to be going slow through there. And as the veins get larger, the velocity increases. So here the veins are getting larger, velocity goes back up because the resistance is going down. The resistance in the capillaries is huge because they're so tiny. As the veins get bigger, the resistance goes down and the velocity or the speed goes back up. Mean arterial pressure is a calculated average pressure. The pressure in the blood vessels is constantly going up and down. Every time the heart beats, it jumps up, and then when the heart's relaxing, it goes down. And it's able to maintain this due to the elastic tissue that's found in the blood vessels that we learned about in the last chapter. So the arteries have lots of elastic in them, and when the heart contracts, it pushes a lot of blood out into them, and that elastic expands. And then while the heart is relaxing, that elastic tissue is recoiling and keeping the pressure up. So the mean arterial pressure, and there's the formula, it's the sum of the diastolic pressure plus 1/3 of the pulse pressure, which is the systolic minus the diastolic. So the average pressure is closer to the diastolic, which is when the heart is relaxed. Next, we're going to talk about capillary hydrostatic pressure. This is due to blood pressure. So there's pressure in the blood vessels, and it drives the filtration at the capillaries. So it's going to push things out of the capillaries. Small things, really small things, can go through the cells. Larger things can go in between the cells. So capillary exchange involves diffusion, filtration, and reabsorption. Small molecules will diffuse across capillary walls and down their concentration gradients, and so they'll go in or out of the blood depending on their concentration gradients. Capillary hydrostatic pressure is going to push water out of the capillaries, and blood colloid osmotic pressure is due to the proteins in the blood, and it's going to pull water into the capillaries. And depending on what part of the capillary you're at, those pressures change. So at the arterial end of a capillary, this is where the blood pressure is the highest. So the hydrostatic pressure at this point is higher than the blood colloid osmotic pressure, and water and solutes leave the capillary. So they're going to leave the capillary and go out into the tissues. In the middle, the pressures are equal and there's going to be no net fluid movement. It's not actually in the middle, it's more like it's closer to the venule end. Then as we get closer to the venule, now the blood osmotic pressure is higher than the capillary hydrostatic pressure. Or we would say the hydrostatic pressure is lower than osmotic pressure, and water and solutes are going to be entering the capillary. Blood flow is regulated by autoregulation and central regulation. If there's inadequate blood flow to an area, the blood vessels leading to that area are going to dilate. So that's going to decrease the resistance and hopefully increase the blood flow and hopefully get us back to homeostasis. If this does not work, then the endocrine and the nervous systems get involved, and they'll try to increase blood volume and blood pressure. Baroreceptor reflexes, these are pressure receptors. They're activated when blood pressure is disturbed. So normal blood pressure is restored by changing heart rate or by causing vasodilation or vasoconstriction of blood vessels. So basically, if the blood pressure gets too high, we're going to dilate the blood vessels. That's going to decrease the resistance and get our pressure back to normal. If the blood pressure falls, we're going to constrict our blood vessels and increase our cardiac output and hopefully get our blood pressure back to normal. When blood pressure falls, the sympathetic nervous system is activated. It increases cardiac output, so we're going to increase cardiac output. It also increases vasoconstriction, so that's going to make the blood vessels smaller, increasing the resistance, and that's going to increase blood pressure. Also, the endocrine system gets involved, and it's going to release EPO, which is going to cause the formation of blood cells. It's also going to release other hormones that are going to cause water to be reabsorbed, and these together are going to increase blood volume. And when we increase blood volume, we're going to increase blood pressure. So that's our response to a declining blood pressure and blood volume. If our blood pressure increases, the heart releases a chemical called ANP, atrial natriuretic peptide. This causes fluid loss from the blood, so it sends more of the water from our blood into our urine, reduces our thirst. It causes vasodilation, so we dilate our blood vessels, making them bigger, reducing the resistance, and all these things combine to reduce blood pressure and hopefully get it back to normal. We also have some chemoreceptor reflexes influencing blood flow and blood pressure. These chemoreceptor reflexes, they respond to changes in carbon dioxide, oxygen, or pH levels in the blood and in the cerebrospinal fluid. So we have some of these in our carotid arteries. Also, we have some in the brain. So when carbon dioxide increases, that's bad. Or when oxygen or pH levels decrease, that's also bad. The respiratory rate is going to increase to bring in more oxygen, and cardiac output is going to increase. And together this will decrease the amount of carbon dioxide. It will also increase our oxygen and our pH levels and bring the blood back to homeostasis. This shows how cardiac output changes with exercise. At rest, we have about 1200 milliliters per minute going to our skeletal muscles. That can increase over 10 times when we're doing heavy exercise. Exercise causes an increased blood flow to muscles and skin. You can see from skin it went to from 500 to 1500 to 1900, and that's because as we exercise we get hot, and when we get hot we send more blood to our skin so we can lose more heat to the environment. So when we exercise, more blood flow is sent to the muscles and the skin. We decrease blood flow to our digestive organs and other organs. Only blood flow to the brain remains constant. See, the brain is getting the same amount no matter what we're doing because we don't want to take blood away from our brain. It's important. Alright, let's talk about some responses to blood loss. The short-term response to blood loss includes increasing the cardiac output and constricting blood vessels to increase blood pressure. The long-term response involves hormones such as ADH, angiotensin, EPO. Those are all going to increase blood volume by reabsorbing water or producing new blood cells, and that's going to work to bring our blood pressure back to normal. If those don't work, if our blood pressure falls, we can go into shock. Shock develops when normal compensation mechanisms cannot maintain adequate cardiac output and blood flow. Circulatory shock. Circulatory shock involves several positive feedback loops that decrease blood volume. Progressive shock involves positive feedback loops, and we can see those here. So we decrease the cardiac blood flow, which causes damage to the heart muscle, which decreases cardiac output, which decreases cardiac blood flow, which causes more damage to the heart muscle, which decreases cardiac output, and you can see that goes on. But we can overcome these usually. But if we don't, these positive feedback loops, they accelerate tissue damage, and they can lead to irreversible shock, which causes circulatory collapse and death. And that is the heart chapter.